Welcome, MS translators, for our Day 2 summary from ECTRIMS in Stockholm, Sweden. After our Day 1 summary ended up going for about 25 or 26 minutes, we've decided that we're going to try and keep this briefer today, although Travis is still planning on talking, so I can't make any guarantees about us actually holding to that promise. So we thought we'd start today by talking about mesenchymal stem cells, because we mentioned those in our Day 1 summary about a trial that didn't work, didn't meet any of its endpoints, but you were in a session this morning that had a different mesenchymal stem cell trial and very different results. Do you want to update everyone on that?
Yeah, so Dr. Crucis, he presented on his findings in a large blinded multi-site trial using very similar approaches, main difference being that they also included a intrathecal delivery. It really struck me how much more robust the data was, and really changed a lot of my perception of the field. And we ended up tracking him down, and I'm sure you're going to be putting it up later. We sat him down for an interview to really dig deeper into what was going on there. But it's definitely worth noting that Day 2 changed a lot of my perspective on the topic.
And I guess that's one of the really interesting points about being here and hearing multiple sclerosis research, and we talk about this a lot, is that it's not an easy disease to work in, and results can be variable. And it was something that you talked about at length with him, trying to explain those differences in the trial results. Also interesting for people probably is that his trial was solely using people with progressive MS. That's correct?
Yeah, that was a big emphasis of today. As I think a lot of people really focus on relapsing remitting MS because the bar is so much higher for the progressive forms of disease, and today really focused a lot more on progressive forms and different clinical trials and things that have been used to try to slow disease progression or get the disease to go back into kind of a senescent state. And obviously a lot of those trials have been negative, but there was a lot of stuff today that showed promise there, and that kind of gets into one of the topics that we want to talk about, because trial design is a big part of this, and the two main emphases that I kind of participated in today was, one, the kind of progressive disease modification, which you did some as well, and then a lot of clinical trial stuff. For my work at Novoron in developing remyelination therapies, trying to learn from past trial failures, what worked, what didn't, to kind of start strategically thinking about what I think the best trial design is going to be for our therapeutic, I think is critically important. I think in a disease like multiple sclerosis, where there is no remyelination therapies, the worst thing in the world that could happen is a false negative, or in other words, a drug that could work or does work, but your trial doesn't show it, and the drug never makes it to the clinic. So there was a lot of really great presentations today on what have we learned from trials. There's a lot of great presentations on just going through and slogging through all the different trials that have been done, particularly in progressive forms of disease. And it was super fascinating. It made me more optimistic about more drugs potentially being approved in the progressive forms of disease, both new and potentially older. But it also gave a lot of really good context for what can be done going forward in trials that can help us better tease out efficacy. Yeah,
and I think two important points made there for the people watching at home. We obviously talk a lot. First point to emphasize that I think people will be pleased about is knowing that, as we've just said, progressive MS is definitely a focus of the conference. There is a lot of people now working on solutions, treatment options for people living with progressive MS. So I know a lot of people have mentioned before that they think that they get forgotten about once they get to the progressive stage, but I can guarantee that's not the case. And also I think the other point that you emphasized there is that I guess a lot of people don't realize why everyone travels to these conferences, and it's not just so that we can have a holiday in Europe. It really is a learning experience for people to come and hear what everyone else is doing, but also to have these discussions when everyone's all in one place, so that we can actually try and get solutions to a lot of these problems quicker. and get outcomes that are going to be beneficial for people living with MS quicker.
Yeah, I mean, it's not very often that you can hear about an exciting new trial and grab the guy out of the room and sit him down for an interview.
Yeah, exactly. And I mean, I've had other meetings today with researchers from Cambridge, talked to some people who are working on the MS Brain Health Initiative. You know, it's these sort of interactions that we only get by being here and talking about these things.
Well,
now,
along those lines, you know, is my role with Novoron. There were pharmaceutical companies that realized I was here, and I was able to have meetings with them about our work. And that's, again, you know, it just shows, like, not only on the science communication or the scientific research or the pharma side, you get everybody in the room, clinicians, researchers, drug developers, and you were able to have this dynamic and you know, diverse interactions, and it's super valuable. It's super valuable, and honestly, it's super valuable here in Europe, because I think a lot of people fixate on the U.S. FDA system, and it's a different mindset in these kind of European social medicine areas, and it's really important to think about those areas, because a lot can be done. There's positives and negatives. There's flexibilities. There's rigidities. But at the end of the day, getting the best treatments to patients requires understanding all of it.
Yeah, no, definitely. So I guess we've just touched upon it. Now, before we did this video and our brief little chat while we were caffeinating up to give ourselves the energy to communicate this through,
chugging those Red Bulls,
Travis said to me, I'm going to spend, and this is a direct quote, I'm going to spend a bit of time riffing about clinical trials. Now, I've told him that he has to keep it maximum 3 minutes to try and keep this video down. But so I'm going to throw to you now, in terms of the clinical trial design, you said it was a big focus today. What are some of the things that are being discussed as being important for that? And your own thoughts, because I mean, it's important, you know, you're here and you are an expert in the field as well.
So, I mean, it's tough to really bring together in 3 minutes with specifics. So what I will say is, it was super revealing to have somebody download 20, 30 of these really well done large trials and be able to compare and contrast and sometimes even combine the data and show what's kind of being revealed, but also just be able to have people honestly get up and stand up and say, this was the limitation, this is what might have been missed. You know, there was a presentation where it was a pretty advanced presentation on the statistics of trial, but the first presenter was like, these are the simple things you have to think of. If you're using an Excel document, save it in the cloud, not on a hard drive. If you use abbreviations, make sure that you have a Word document that has clear explanations of what those abbreviations are. And she was saying this as somebody who runs the clinical trial or runs the multiple sclerosis kind of portfolio, I think for Spain, and I don't want to be wrong about that, but she's like the national person who kind of observes these things. And she's hammering home these simple things because people get it wrong. And it's important. It really drives home how these simple things are really important to emphasize on. And then on top of that, then you start getting into things like, you know, cross cohorts versus, you know, it's really valuable to see those perspectives, to see the emphasis and learn in this situation, because I really do think in multiple sclerosis, the thing that we can't afford to do is take shortcuts or try to be, conservative in the way we approach things and risk a false negative, because you might lose the opportunity to show that a drug works that could really help people.
Yeah, and I think that's, I mean, when we talk about these things, and obviously there is an understanding that as an MS community, we want answers quickly, we want improvements quickly, but certainly when we're talking about clinical trials, spending a little bit more time to get the trial design right, to give the trial the most chance of success, is really important, because what we're going to waste far more time if we start running trials, putting participants into these trials that are destined for failure from the beginning just because the trial hasn't been designed in a way that gives it any chance of success. So I think that's why these conversations are really important, and we get these things right, so that we do end up getting the answers that we're looking for.
And it's also really refreshing, because a lot of times trial designs, you hear about the negatives as from a research perspective from the decisions that pharma made. And it's really easy to nitpick pharma. It's low-hanging fruit for many reasons, but it's really interesting to hear at this conference the physicians explain some of the rationale that you can easily write off as short-sighted or misguided, and hear the explanation, and you're like, oh, I get it. You can learn from a mistake. It wasn't just a horrible idea, right?
Yeah.
So it's a lot of times you learn more from the failures, and you learn more from the context of what led to that decision than you do from quarterback couching and trying to nitpick on what you can obviously see in hindsight, right?
Yeah.
So,
and I mean, that was one of the really interesting things. And as we said, we'll publish this interview, the full interview, soon. But this
it was a good one. By the way, my first interview. So if it's horrible, it's Brett's fault.
He did a good job. But what really came out was we had the trial yesterday that didn't work. We had the trial today that worked, and comparing them gives us some really clear answers as to the best ways that we should be doing this when we're looking at this sort of treatment as an option.
Yeah.
So I went to a couple of other sessions that were quite different in terms of their output. One was looking at symptomatic treatments, and this is something that I touched upon last year as being something new in the field. That we're now starting to, while there's an understanding that we need to be looking into how best to modify the disease and treat the disease and stop new attacks, that at the same time, if we want to be sort of improving the daily quality of life for people living with MS, we also need to be treating some of these symptoms. So, what is the best way that we can actually just treat fatigue? That treatment for that may not be doing anything for the underlying disease process, but it doesn't mean that it's not important. And so there was talks about that today. Probably the most interesting one that we've touched upon before, and as you've said, it's being used now across a variety of different diseases, is looking at transcranial. Now I'm forgetting what the
direct magnetic stimulation,
direct electrical stimulation.
It would be transcranial. Okay.
Direct current stimulation.
Yeah.
There we go. It's been a long day. Essentially giving little electrical impulses to the brain. And they found that this can help in a variety of different symptoms, but interestingly what they talked about today was that the treatment alone, you might get a little bit of an effect, but really it has to be done in combination with some sort of training.
Yep.
So if this is being done, say, to improve spasticity in the legs, just doing the stimulation may get some sort of benefit, but when combined with exercise, that's improved greatly.
And just to touch on that real quickly, I think that's something that in neurology has only now recently started to get more traction. I mean, the spinal cord injury is a similar thing. A lot of our preclinical work is just injuring the spinal cord and seeing what happens. And I think people are starting to realize that the efficacy of an intervention, a drug or a therapy, is not going to be as powerful if you're not actively. It's like rehabbing a surgically repaired knee. Like you might mechanically make that sound, but if you're not strengthening things around it and relearning how to use it, you're not going to get the same use out of it later. And I think for a long time we took that for granted, and it's the data is starting to realize it. Just like with that, like you have an intervention that does things, but it's so much more impactful when you train with it, right?
Yeah. And so they've shown similar things with using it to improve cognition. Stimulation alone, no. Combine it with some sort of cognitive brain training, much better results. So that was one thing. The other thing was comorbidities, and I think the most interesting thing from that was looking at the MS prodrome. So essentially, all of the things that are happening before people are being diagnosed with MS. And one of the interesting study results from that was that there was clear evidence from one of the registries in Germany that showed that people with MS were more frequently going to a doctor than the general population, and often for psychiatric and neurological complaints, even years before being diagnosed with MS. So
it's kind of like the Parkinson's space.
Yeah, and so the sort of outcomes of this may be that if we can see these sort of red flags much earlier, we know that the earlier we can diagnose, the earlier we can get people on a highly effective therapy, the better the long-term outcomes. So being able to identify those red flags, really important.
Yeah, it's interesting. You know, that's one of the nice things about big data now is we can collect this information in a way we couldn't before.
Yeah.
You know, like in Parkinson's, there was anecdotal evidence for a long time that the sense of smell would start to be altered before any movement disorders actually took place, and
Yeah.
and now there's a whole symptom profile that people are starting to think was really standard before the kind of more, you know, dogmatic movement disorders come into play. And I hadn't heard that about MS, but you know, the reality is it doesn't surprise me.
Yeah.
So that's really interesting to hear.
Well, and as you say, I think this, to sort of finish off today, that comment is really meaningful, because part of the reason why we talk to you guys about research is that we also want to hear from you guys about your experiences, because they can tie in with everything that's happening here. And that anecdotal evidence that you talk about, when we actually hear from people living with MS, that can help shape research directions. And that's what we're starting to see now. So that's our day 2 wrap, hopefully a little bit shorter than our day 1 wrap.
I know, I can never tell.
Yeah, we'll see when we get to the end of it. We're excited to present day 3 as well. There's some poster sessions that we're going to now. We may have more information on that tomorrow. But thank you for watching, as always. Thank you, Travis, for being involved. And we'll talk to you tomorrow. Thanks, everyone.
Well, it didn't